right, so the last we left off, we have transformed our bacteria and plated them onto our LB agar plates with kanamycin. Typically, you would allow these plates to incubate at 37 degrees overnight. If you're going to need to wait longer than that, you need to come in after the overnight incubation and at least put those plates at 4 degrees in the refrigerator until you're able to do the next step. So I wasn't able to come in right the next morning after I plated these bacteria, so I arranged for a lab mate to place these plates in the fridge at 4 degrees. And what I saw when I came in is that the plates did have colonies. So you can see some of the plates have quite a lot of colonies. You can see little growths of bacteria. And then some of them had colonies, but just a few, not a whole lot. So this one just had a handful. Really all you need is one some of the time. This one has more like 4 or 5. So if you're not able to start these mini prep cultures right away, like maybe you do this on a day where you can't come in the next day, you can put those at 4 degrees in the refrigerator, or have a lab mate do that for you. Once you're ready to do the next step, you're going to set up a culture overnight, and then the following day you're going to come in to do what's called a mini prep to purify the plasmid from the bacterial culture so that we can send it to sequencing. So what I'm going to do today is to set up those cultures. And the first thing I have is I have my plates out. I've taken them out of the fridge. I have my culture tubes. So these are some Fisher brand culture tubes that look like this. They have a little lid. And I've labeled them with numbers for each one of my samples. And the next thing I'm going to do is get some LB liquid media with kanamycin. And this is in the refrigerator. So this is our BWR refrigerator. And there is a bottle in the back of LB media plus kanamycin. So try not to get you dizzy, but we'll take this back to our bench. We're going to use our pipetter to pipette 5 microliters of liquid media into each tube. So we'll do that first. If you haven't labeled your tube, you can go ahead and pause the video. And I'm going to pipette 5 milliliters into each tube. And these big pipettes can go in our special plastic waste for the taste. So each tube will have 5 milliliters of liquid media with LB plus kanamycin. The kanamycin is there just to maintain selection of the plasmids. Now what I'm going to do is I'm going to take my P200 tips. I'm going to carefully remove one of those tips without touching any of the other tips. Just hold it in my hand. And I'm going to look at my first plate and I'm going to just barely touch this onto one of the colonies. You don't want to gouge the plate, but you do want to get that colony of bacteria. And then I dropped it into my culture tube. Then I can put the lid on. So overnight, those bacteria from that colony are going to grow into a liquid media that we can purify the plasmid from. So here again, this is what this looks like. I'm just going to, I don't know if I can try to do it this way. Just going to choose one of those single colonies, make sure that you place it in the right tube, and do that again for the others. So you don't want to gouge the plate, but you do want to get one of those colonies. It's also important that you only get one colony, not several of them, because each colony represents one transformant. And you could have transformants with different plasmids in them. So you really want to specifically get one of the colonies, only one, to place into your culture tube to grow up. So what we'll do is place these in the incubator and let these grow overnight. So I will take you with me. Just turning on these lights. This is our shaking incubator. And I'm going to place, there's a blue culture tube rack back here, so I'll just place them in here. It's harder to do one-handed. And then I'm going to place this on, make sure that the power is on, and you should have both the shaker and the heater on so that the shaker can grow the bacteria. So once you put the bacteria in the shaking incubator overnight, this will encourage the culture bacteria to grow so that you can purify the plasmid from it. From here, what you would do is just put the liquid media and your bacterial plates back in the refrigerator for safe storage. And then that's it for the evening. You only want to let these cultures grow for about 16 to 18 hours, so you want to plan accordingly. You don't want to set up cultures at 10 o'clock in the morning, because then by the time you came in the next day it would be too late. The cultures would be overgrown. So for example, if you wanted to start them at 9 in the morning, you could set them up just last thing in the evening before you left. So that will be our next step, and then after that we will have our plasmids to send for sequencing to see if our cloning worked. That's it for today. So today we're going to resume purifying the plasmids that we've grown in the bacteria from our pre-culture. So we've grown these 5 milliliter cultures overnight. You can see that the bacteria have grown, so the solution is now cloudy. And what we're going to do today is do what's called a mini prep. And this allows us to purify the plasmid, our PJC plasmid, which hopefully contains our inserts from these bacteria, so that we can send it for sequencing and so that we can use it as a template for other protocols in the lab. So there's a kit called the Zippy Plasmid Mini Prep Kit. It actually lives on that shelf right there. And this is a kit that allows us to do this mini prep that we're going to do. It has a set of instructions in the box. There's also one of these on the end of my bench by the centrifuge. And you can also find this online if you want your own personal copy. In this box there are a few solutions. There is this blue 7X lysis buffer. There's actually a couple of these since that one's almost empty. There is an endo wash buffer. There's the Zippy wash buffer, and this is one of those that needs to have ethanol added, so double check that first. And there's the elution buffer. There's also another buffer that's yellow called the neutralization buffer that's in the refrigerator, and that needs to stay refrigerated the whole time. And before I start, I have labeled a set of 3 sets of tubes. The first is just a regular 1.5 ml tube. This is where we're going to lyse the bacteria. The second is I have a set of collection tubes with the columns in them. And these columns are a little bit different than the columns that are used to purify DNA just from a solution. So we have 2 different types of columns. Once you see each of them, you can see that the filter on the mini prep column is wider than the one that's in the other kit. So if you ever take one out and you think, oh, this doesn't look right, just make sure you have the right columns for what you're trying to do. So this together, this collection tube and the column, is what you need for your second set. So I've labeled my first set of tubes just 1 through 4 because I have 4 tubes. I've labeled the sides of these columns 1 through 4. And then I've labeled another set of 1.5 ml tubes, and this is where I will actually have my mini preps. So I have MP for mini prep 1, I have the date, you can put your initials on the side, other information. But these are ones that you're going to keep for a while, so you want them to be really well labeled. So the first step of this is to, like I said, make sure that this buffer has the ethanol added. It should, but if not, you can get some help with that. And the second thing is that if we have this 5 milliliter culture, we can't fit that into our 1.5 milliliter tube. So the first thing we're going to do is go and centrifuge these bacteria down so that we can put them in a smaller volume. So what I'm going to do is go over to this centrifuge that's by the microwave. This is it right here. And this centrifuge can be used for larger volumes. So right now it's set for just this kind of culture tube I have. So I can put these tubes in, make sure that they're balanced, meaning, you know, you need an even number of tubes, or you need to have one tube with just water in it. So there, I've made sure that these tubes are set apart. If you ever need a different type of tube, you come here and those cups are not in there. There's some other tubes, tube racks and things like that at the bottom that you can use. So I'm going to set this centrifuge at, let's see if I can get down here, 25 degrees Celsius at 3,000 RPMs and for 5 minutes. And then I'm just going to hit start. And so we're going to let that go for 5 minutes for our spin, and then we'll resume for the next step of the protocol. Okay, so the spin is done, and you'll now see that the bacteria are in a pellet on the bottom of the tube, and the liquid has been kind of cleared as all those bacteria have been pulled down to the bottom. And so while that spin was going on, I grabbed a biohazard bucket and also this vacuum flask, which I'm now going to turn on. And I'm going to put a P200 tip into the end of this that has a syringe into it. And so what I'm going to do, I'm actually going to also grab a transfer pipette that I'm not actually going to use per se, but what I'm going to do is uncap the first tube, use the transfer pipette just to remove the tip that we use to inoculate the culture into the biohazard bag. And I'm now going to aspirate the media into the vacuum flask, being careful not to disturb the pellet. So I'm just going to take this vacuum and go down the side of the tube, removing some of the media at a time until I get right to the edge of the pellet. What's left in here is the bacterial pellet and no media. So let's do that with one more of these samples so you can see. I'm going to remove the cap. Going to take out that tip. You can either use a separate one of these for each one, or I'm just sitting it on a rack like this so it doesn't contaminate anything, and then I'll throw it in the biohazard too when I'm finished, and then using the vacuum flask again to remove the liquid medium. So again, have a pellet. So I'm going to do that with the other two tubes that I have, and then we'll move on to the next step. Okay, so next we're going to lyse these bacteria. We have the bacteria in the tube as a pellet, and first we're going to move this bacteria into the 1.5 ml tubes. This protocol says you need to have 600 microliters of bacterial culture grown in LB medium. And so what we're going to do is set our pipetter to 500 microliters with the expectation that these pellets have some volume of their own. So together the 500 microliters plus the pellet will be about right. So I'm going to open my first set of 1.5 ml tubes and pipette just some sterile water into the first bacterial tube. So I'm going to pipette this in here. I'm going to pipette up and down and stir to resuspend these bacteria. And then I'm going to pipette this into my first tube. Make sure you're being careful about which bacteria go into which tube. I'm going to eject this tip into this culture tube and put the cap back on it. And then that will be ready to just go straight into the biohazard when you're finished with this step. So again, we're going to add water to the second tube. Same thing. And since you have a little bit more liquid in there than 500 microliters, it usually takes 2 pipettings to get that material out. So we're adding 500 microliters, resuspending, pulling out that 500 microliters, and then pulling out the rest. And that's going into these this first set of 1.5 ml tubes. So I have one more tube. Okay, so those can just go into my biohazard bag. Okay, so according to the protocol, the next step will be to add 100 microliters of the lysis buffer. For these protocols, I'm using the sterile tips, not the RNA-free tips. This isn't RNA work, so you can just use the regular P200 tips. And if you're really careful and don't cross-contaminate, you can use one of these tips, but if you worry about touching anything, you can use a separate tip for each tube. I've probably pipetted a million times in my life, so I can be pretty careful at this point. And then this is not biohazard waste. This can just go in your regular tip waste tube. So you're going to cap your tubes, and you'll see the 7X lysis buffer looks like this. So I'm going to invert these tubes several times to make sure that the solution is mixed well. And then as per the protocol, it says to incubate for 1 to 2 minutes. And so I'm just going to start a timer. This is something where you don't want to let this go too long over. And so if you had 20 tubes, you might do this in sets of 10 at first or something like that. I just have 4, so I'm doing them all together. But if you had a lot of them, you might do the lysis step in 2 groups. So it also says do not vortex, so that's why I'm just kind of gently inverting these. We're trying to purify DNA and we want it to be intact. So we don't want to do anything like pipetting up and down or vortexing that could create shear forces that damage the nucleic acids. Okay, in the meantime, I also got out this bottle of neutralization buffer from the refrigerator. It's yellow. There's a pH indicator in this, so what we'll expect is that once we add that to this buffer that's blue, it will take on this yellow color, and we'll also see precipitation of a lot of the materials from the lysed bacteria. So this is the bacteria lysing in this sample, and the neutralization buffer will stop that, neutralize the pH, and cause precipitation of all the cell wall material and proteins and other things like that. And it's important that this neutralization buffer is cold, so you should only get it out of the fridge right before you use it, and then put it back in the fridge right after you're finished. So this works best with that neutralization buffer cold. So it's about 1.5 minutes, so I'm going to uncap my tubes and add 350 microliters of cold neutralization buffer to each tube. And again, if you need to use a different tip for each sample, you can do that. If you can make sure not to, like there, I touched the side a little bit, so I'm going to get a new one. If you can avoid touching anything or splashing anything on your tip, you could use the same one. And then again, this is a reminder, we keep all of our tips separate so that we can put them in the plastic waste so they don't puncture the trash cans. So when you add the yellow solution, you can start to see some mixture of this is not very good at first, but I actually just kind of shake these a little bit more rigorously than I shake the first step. And you'll see at first there might be some little blue blobs still in there, but as you shake more, the tube will have a solution that's completely yellow. The other thing you will see is it'll look kind of chunky, so it'll have an opaque, sort of flaky substance in the tubes. Looks like a little bit like a liquidy scrambled egg or something like that. So there you can kind of see the chunky stuff in there. So what we're going to do first is I'm going to put this back in the refrigerator, and then we're going to spin these tubes. As per the protocol, it says centrifuge for 2 to 4 minutes at something greater than 11,000 G. And so I will take you with me. just going to turn the centrifuge on. I'm going to put this bottle away, just right here. And then we're going to set the centrifuge to 4 minutes and keep it at room temperature. And we want this speed to be something over 11,000 Gs, so make sure this is set to Gs. We'll do 12,000. Okay. So I'm going to put my tubes into this centrifuge. Remember to keep your tube balanced. So the 2 tubes, since I have 4, will be opposite to the 2 other tubes. And make sure you have the lid on carefully. It will close itself, and then we'll just hit start. Okay, so that's going to spin, and what's going to happen during this spin is that the chunky cell lysate will pellet to the bottom of the tube, and the clear liquid that will contain our plasmid will be toward the top of the tube. And I'll show you what that looks like, and we'll move forward. All right, so the spin has completed. I'm going to show you what this looks like. I'm just putting these tubes into my rack really quickly. Very gently, you don't want to disturb the pellets. So what these will look like would be this. You see the chunky cell debris at the bottom, and you see a clear supernatant at the top. What you're going to do with each one of these is carefully uncap the tube and pour this liquid into the column. And you might get a little of that chunky stuff in there, but you don't want to get too much. So I leave a little bit of the liquid in there rather than trying to get it all out and dislodging that cell debris in the tube. So we'll just show you another one of these. It'll be like that. And I like to let those sit for maybe 30 seconds, and then you're going to do another spin. So the tube that has the cell debris can go into the biohazard. And then we're going to spin those again for I'm just checking. It says 15 seconds. We can't actually do that with the centrifuge, so you can do 30 seconds. So after the spin, you'll see that the supernatant has gone through to the bottom of the tube. I'm just going to go dump this out in the sink. I'm going to put these tubes back into my rack. And I brought over to the centrifuge the 2 wash buffers. First the endo wash buffer, and then the zippy wash buffer, and some tips in my pipette. So the first wash is with 200 microliters of endo wash buffer. I'm going to just add this to each. I'm going to just pipette this, add this to each column, being careful not to touch anything to contaminate my pipette. Going to centrifuge this again for 30 seconds. Okay, and after that first spin, you can see that the liquid is through. You can dump it out after this point, but you can actually fit both of these washes in there without dumping it between them. So next I'm going to add 400 microliters of the zippy wash buffer. So these 2 washes will get any remaining materials off of that filter besides the DNA, and leave you with a pure plasmid. So I'm going to put that back in and spin for 1 minute. So after that spin, we have both of those washes through, and I'm going to just dump that out into the sink. You can see here, this is why I label the column instead of the collection tube, because you're dumping it out a lot of times so you can get them mixed up if you don't label the column. I'm actually going to put the tubes back in the centrifuge and spin for 1 more minute. That is a drying spin, so any liquid that's remaining in the columns will be washed off, and some of the ethanol may even evaporate. After the drying spin, you want to take these columns and collection tubes out. Do not empty these collection tubes, because if you did, what would happen is you would pour out the liquid, put the column back in, and then the buffer or ethanol would get back onto the column. So I'm just going to take these out and put them on into my rack in order, making sure they're in the same order as my labeled tubes for the elution. And then I'm just going to lift these columns out and place them into the tubes that I want to elute into. And so in that sense, these collection tubes, you don't really need to empty them because they're trash next anyway. So what I will do is I will actually let these tubes dry for 1 minute, and then we will elute the plasmid. And our last step is the elution. There's an elution buffer in the kit, and this is a little bit different from water in that it has some other buffer elements in it. So this will make your plasmid DNA stable in the freezer over a long period of time. So as per the protocol, we're going to elute in 30 microliters of this elution buffer. So we'll get our pipette and pipette 30 microliters of this elution buffer onto each filter. And then I also let this sit for about a minute before I do the centrifugation step. This allows time for that buffer to sit with the DNA that's on the filter and kind of resuspend it off of the filter before centrifugation happens. So both this 1 minute drying step and this 1 minute of incubation before you do the final spin can help you with that, with kind of maximizing your yield. So I'm going to centrifuge these 1 more time for 30 seconds, and then we will have our DNA. So once you've taken the concentration of each plasmid, you need to prepare samples for sequencing. I'm not going to go into a whole lot of detail about this because it's possible that down the road we will change the way we do this. But for right now, the way we do this is we send our samples to GeneWiz, which is a company that can do Sanger sequencing. And to prepare samples for sequencing, you want to make dilutions of your plasmids in PCR tubes. So for example, I have 4 samples. I'm going to prepare those 4 samples in a strip of 8 PCR tubes. I'm going to write on them, my initials are RG1, 2, 3, 4. So you don't want to just label them 1, 2, 3, 4 because other people could have samples with the same name. So I've just labeled the first 4 tubes in this strip. And I've also calculated how much of the sample I need to add and how much water I need to add. So for each one of these tubes that you want to send for sequencing, you need 10 microliters, and you need a total of 500 nanograms. So that's, it ends up being 50 nanograms per microliter final concentration and 10 microliters of that. So you'll need to do the conversion to figure out how many microliters of your Miniprep sample you need to add and how many microliters of water you need to add to that to have a final concentration of 50 nanograms per microliter and a total volume of 10 microliters. So I'll give you my first example is a Miniprep with 390 nanograms per microliter as the concentration. And when I do the math for this, I have to combine 1.3 microliters of that plasmid with 8.7 microliters of sterile water, and that would give me 10 microliters of 50 nanograms per microliter. So I'm going to combine those water and Miniprep into these 4 tubes, and then I'm going to send them for sequencing. If you're an undergraduate or a rotation student, you'll probably work with someone who's already in the lab to send these samples for sequencing. If you join the lab, we should set up an account so that you can start sending your own sequences, and you can work with one of those when you get to that point. So I'm going to set up my samples. I'm going to add the water and solution. I'm going to put a strip of caps on here, and I'm going to wrap it once with parafilm, which is a kind of a waxy paper. And after that, I'm going to send these for sequencing. When I get the sequences back, we'll evaluate the sequences to see if the cloning worked. So this is what the samples will like when you wrap them up with parafilm. That's just to keep the samples from evaporating or popping open in transit. Your Minipreps can go into your freezer for storage, and then we'll evaluate the samples when we get the sequencing back. One last item on this: after you finish the Minipreps, you want to make sure everything is back in the kit and put it back on the shelf, with the exception of the neutralization buffer, which you should have put back in the fridge when you were finished with it. If you haven't, put it back now. You want to clean up the vacuum flask with bleach and make sure that it's decontaminated, since it's had contact with bacteria, and make sure that all of your tubes and stuff that have come into contact with the bacteria have gone into the biohazard waste. So that's kind of the housekeeping stuff to finish up the Minipreps.